Hello students and my dear friends, myself Madhu Vancha. In this session, we'll discuss some of the important MCQs which will be helpful in the lab technician recruitment examination. First question, what is the sputum black color clinical condition? Which means in which clinical condition we can see the like a black color sputum. Option A, due to inhalation of coal dust. Option B, pus. Option C, epithelial cells. Option D, necrosis. Here the right answer is option A. That is due to inhalation of coal dust. So here's sputum black color condition. It can be because of the due to inhalation of coal dust. So this is, you can see in the coal workers, those who are having pneumoconiosis, or even we can see in case of heavy smokers. Next question, what is the cell nuclei color in hematoxylin and eosin staining method, in H&E staining method? Option A, red color. Option B, green color. Option C, yellow color. Option D, blue color. Here the right answer is option D, blue color. So in the hematoxylin and eosin stain, so hematoxylin, it stains the nucleus and eosin stains the cytoplasm. So now in this staining, nuclei appears dark blue in the color and cytoplasm appears in varying shades of the pink color. So cytoplasm pink and nuclei appears in dark blue color. Here the right answer is blue color. Next question, fixation treatment of tissues may cause. Option A, increased rate of penetration. Option B, decreased rate of penetration. Option C, both. Option D, neither. Here the right answer is option C, both. So what is meant by fixation? It is the process by which tissues are preserved as natural state as possible. The tissues are preserved as like a natural state as possible, that we prevent both autolysis and putrefaction. So here fixation of treatment causes either it may cause increased rate of penetration or decreased rate of penetration. Next question, what is histopathology? What is meant by histopathology? Option A, study of diseased tissue. Option B, study of functions. Option C, study of cell division. Option D, study of shapes. Here the right answer is option A, study of diseased tissues. So if you see the word histopathology, it is the Greek word which means like histos means tissue, patho means suffering, and logy means study. So histopathology, it involves the diagnosis and study of diseases of the tissues. It is the study of the diseased tissue, and it involves the examining tissues or cells under the microscope. Next question, pale yellow color appearance of peritoneal fluid indicates which clinical significance? So pale yellow color peritoneal fluid indicates which clinical condition? Option A, hepatic vein obstruction. Option B, cirrhosis. Option C, nephrotic syndrome. Option D, all of the above. So if you see, the right answer is option D, all of the above. And peritoneal fluid, generally it is clear. So the color is like a clear and straw. So what is the normal volume of peritoneal fluid? That is around 50 ml. So here in case of pathological conditions, in case of hepatic vein obstruction or cirrhosis or nephrotic syndrome, so we can see the yellow color peritoneal fluid. Next question, what is the normal pH of sperm? Option A, 7.2 to 8.0. Option B, 7.8 to 9.3. Option C, 4.5 to 6.9. Option D, 3.5 to 7.9. So here if you see, the right answer is option A. The normal pH of sperm, it is alkaline in the nature. It has alkaline pH that is 7.2 to 8.0. So when we should measure the pH of the sperm, it should measure after the liquefaction. So generally after 30 minutes of the collection, we should check the pH of the sperm. Next question, define hyperspermia. So what is meant by hyperspermia? Option A, total absence of ejaculate. Option B, absence of spermatozoa in semen. Option C, a reduction in the volume of the ejaculate. Option D, increase in semen volume. Here the right answer is option D, increase in semen volume. So here what is the normal volume of the semen? That is more than or equal 2 ml per ejaculate. So here the normal volume is more than or equal 2 ml per ejaculate. So here we can see the low semen value that is less than 1.5 ml. It is due to the obstruction of ejaculatory duct or congenital bilateral absence of mass difference, or it may be due to the difficulty in the collection also. So high semen value, so that may be due to the active exudation in cases of inflammatory lesions of accessory organs. And not only that one, in case of infertility also, there is a slight excessive in the semen value. And remember one point, the value of the semen, it doesn't vary with the period of absistence. So if you see other conditions, total absence of ejaculate is called as anejaculation, and absence of spermatozoa in semen called as agiospermia, and reduction in the volume is called as a hypospermia. So hypospermia, that is less than 1.5 ml. Next question, what is the appearance of CSF in bacterial infection? Option A, cloudy. Option B, clear. Option C, cloudy to purulent. Option D, clogged. So here the right answer is option A, cloudy. So if you see the normal CSF is clear and colorless. So in case of bacterial infection, the CSF will be in the cloudy. Next question, what is the normal value of CSF in adults? Option A, 150 ml. Option B, 25 ml. Option C, 300 ml. Option D, 10 ml. Here the right answer is option A, that is 150 ml. So the total volume is in between 120 to 150 ml. So here in these options, there is no 120 to 150. So here we can consider as 150. So here normal CSF volume is 120 to 150 ml. So what is the rate of production of CSF? That is 500 ml per day. That is rate of production. And how much CSF sample is collected? That is 10 to 12 ml under normal pressure. Next question, what are the factors that regulate erythropoiesis? So what is erythropoiesis? That is production of RBC. So what are the factors regulate erythropoiesis? Option A, hypoxia, and option B, hormonal factors, that is ACTH. What is ACTH? Adrenocorticotropic hormone. And next to TSH, that is thyroid stimulating hormone, and thyroid hormones, growth hormones. So all these factors. And option C, vitamins such as vitamin B12, folic acid, and ascorbic acid. And option D, all of the above. So what are the factors regulate erythropoiesis? Here the right answer is all of the above. So environmental factors such as hypoxia, hormonal factors, and vitamins, all these factors, they regulate the erythropoiesis. Next question. Occasionally, structures can be seen within the Cryptosporidium boabum oocyst that are stained with the modified acid fast stain. They are option A, sporozoites, option B, blastocysts, option C, gametocytes, and option D, sporoblasts. So we should know what is Cryptosporidium boabum. So Cryptosporidium boabum, it causes cryptosporidiosis. It causes cryptosporidiosis. This disease was discovered by the scientist Tizer in the year 1907. And if you see oocyst, what is oocyst? The infective form of this parasite is the oocyst. So how this infection takes place? It is by the ingestion of food and water contaminated with feces, which consist of oocyst. And if you see the oocyst, there are two types, thick-walled oocyst and thin-walled oocyst. So both thick-walled oocyst and thin-walled oocyst, they consist of concentric rings. So concentric shaped rings, they are called as the sporozoites. So they are stained by the Jaden staining, that is acid fast staining or modified Gill-Nielsen staining. They appears bright red in the color. Here the right answer is sporozoites. Next question, motile reproductive stage of Entamoeba histolytica is option A, trophozoite, option B, cyst, option C, precyst, option D, none of the above. Here the right answer is option A, trophozoite. So trophozoite, it is a motile form. So it is a motile form. It is motile by the pseudopodia. So what is pseudopodia? It is a long finger-like projection. Next question, a 24-hour urine sample from a man who had no evidence of kidney impairment was sent to the laboratory for hormone determination. The volume of urine was 600 ml. The volume of urine was 600 ml, but there was some question as to the completeness of 24 hours collection. The next step would be option A, perform the hormone determination since say 600 ml of normal urine in 24-hour value. So option B, check the creatinine level. If it is less than 1 gram, then do the procedure. And option C, report the hormone determination in milligrams per deciliter in case of specimen was incomplete. And option D, so check the creatinine level. If it is more than 1, then do the procedure. So here what we should do in this case, here what is the right answer? Option D. So first of all, check the creatinine level. If it is greater than the 1, then do the procedure. Next question, what are the methods of determination of clotting time? Option A, Levite method. Option B, capillary tube method. Option C, EV method. And option D, A and B, that is Levite method and capillary tube method. Here the right answer is option D, that is A and B, which means Levite method and capillary tube method. These two methods are helps to to determine the clotting time. And if you see another option, EV method, that helps to determine the bleeding time. Next question, a positive test for blood in urine can occur in all the following except. So here the blood in the urine. What is blood in the urine? Hematuria. So in which cases we can see the hematuria. So option A, caliculi of the kidney or bladder. Option B, malignancy of the kidney or urinary system. Option C, crush injury or trauma. Option D, extravascular hemolytic anemia. So here the right answer is option D, extravascular hemolytic anemia. So in case of caliculi or in case of malignancy or in case of trauma, we can see the hematuria. All of the statements below about urine color are true except option A, normal color is due to the dietary products and water-soluble bile pigments and to inhalation of the coal dust. And option B, urine may turn dark on standing if high levels of urobilinogen present. Option C, deep yellow urine with yellow foam may contain the bilirubin. And option D, abnormal color is consistently caused by metabolic disease. Here the right answer is option D, abnormal color is consistently caused by metabolic disease. So in case of the A, B, C, so generally it is true, but in case of abnormal color, so that is caused by metabolic disease is false in this case. Next question, What is the use of India ink preparation of CSF? Option A, identification of candida. Option B, identification of histoplasma. Option C, identification of cryptococcus neoformans. Option D, all of the above. Here the right answer is option C, identification of cryptococcus neoformans. So India ink preparation, so that helps to identify the capsule. So cryptococcus neoformans, it causes cryptococcal meningitis. So in case of meningitis only, we collect the CSF sample. So to identify this cryptococcus neoformans, generally we go for India ink staining. So where we will identify the capsule. So the capsule appears as clear halo structure. Next question, what is the normal range of clotting time by Lee-White method? Option A, 5 to 8 minutes. Option B, 7 to 10 minutes. Option C, 4 to 11 minutes. And option D, 8 to 4 minutes. Here, what is the normal clotting time by Lee-White method? Here, the right answer is option C, that is 4 to 11 minutes. Next question, what is the composition of Rees-Ecker fluid? Option A, sodium citrate. Option B, formalin. Option C, brilliant crescent blue. Option D, all of the above. So here, what is Rees-Ecker diluting fluid? So that is a diluting fluid for platelet count. So here, what is the right answer? Option D, all of the above. So here, this fluid consists of sodium citrate, formalin, and a brilliant crescent blue. Next question, what is the osmotic fragility test? Option A, the osmotic fragility test to assess their ability to withstand hypotonic saline without bursting. So here in this, we check the RBC, whether they are able to withstand hypotonic saline without bursting. Or option B, what is that is ability of oxygen transport. And option C, ability of carbon dioxide transport. And option D, estimate the level of HB. So here the right answer is option A. So with the osmotic fragility test, we assess the ability of RBC to withstand hypotonic saline without bursting. Next question, which of the following is characteristic finding on the blood smear of a specimen that has reticulocyte count of 21.5%? Option A, hypochromic macrocytes. Option B, polychromatophilic macrocytes. Option C, basophilic stippling. And option D, oval-Joly bodies. Here, the right answer is option B, that is polychromatophilic macrocytes. Next question, which of the following is an agranulocyte? Option A, neutrophil. Option B, monocyte. Option C, basophil. Option D, eosinophil. Here the right answer is option B, monocytes. So what are agranulocytes? Agranulocytes, they are the type of WBC which has no distinct granules in the cytoplasm. They have no granules in the cytoplasm. And if you see granulocytes, so granulocytes, they are also type of WBC. They have small granules in their cytoplasm. So if you see agranulocytes, what are the agranulocytes? Lymphocytes and monocytes are agranulocytes. What are granulocytes? Basophils, neutrophils, and eosinophils. Next question. A patient has cold agglutinin disease. How can a good blood smear be made from this patient's blood? Option A, dry the smear quickly. Option B, decrease the angle of the spreader's slide. Option C, press hard on the spreader's slide. Option D, warm the blood to the 37 degrees centigrade before making the smear. So here, what is cold agglutinin disease? Cold agglutinin disease, it is the rare autoimmune disease. It is a rare autoimmune disease. So it is characterized by high concentration of circulating cold sensitive antibodies. So there is higher concentration of cold sensitive antibodies. So these antibodies are generally IgM antibodies and autoantibodies. So they are active at a temperature below 30 degrees centigrade. So if the temperature is below 30 degrees centigrade, so generally these antibodies become active and they act against the RBC. So as they act against the RBC, so they agglutinate with the RBC and they undergo the lysis. They undergo the destruction of the RBC. So here in this case, how the blood smear of the patient is prepared. So first, if you collect the blood sample, how the blood smear is prepared in case of cold agglutinin disease. Here the right answer is warm the blood to the 37 degrees centigrade before making the smear. Next question, which diluent is used for manual platelet counts? Option A, 1% phylloxin B. Option B, 3% acetic acid. Option C, 0.85% sodium chloride. Option D, ammonium oxalate. Here the right answer is option D, 1% ammonium oxalate. So 1% ammonium oxalate, it is a diluent which is used for manual platelet count, thrombocyte count. Next question, what are the composition of pilot solution? So what is meant by pilot solution? So pilot solution, it is used for absolute eosinophil count. So here option A, phylloxin B, that is 1% solution in water, that is 10 ml. Option B, propylene glycol, that is 50 ml. Option C, sodium carbonate solution. And option D, all of the above. Here the right answer is option D, all of the above. So as I told you, pilot solution, so it is used for absolute eosinophil count. So in this case, the blood, it is diluted 10 times in a WBC pipette by using of pilot's diluting fluid. And what this pilot's diluting fluid it do? It lyses the RBC and it lyses the leukocytes except eosinophils. So except eosinophils, so it lyses the RBC as well as other WBC. So that is the purpose of the pilot solution. So it helps to check the absolute eosinophil count. So it is helpful to count the check the absolute eosinophil count. Thank you.